Okay, here's part 3 of chapter 7. So we're still talking about proteins in the cell membrane. We talked about facilitated diffusion and how they allow diffusion to occur through the hydrophobic cell membrane. But some proteins actually take it a step further and become carrier proteins because they bind to the molecules that are being transported, and then they change shape, just like an enzyme might, and shuttle the transport molecule across the membrane. Just like enzymes, also, these transport proteins are very specific for the substance that they're actually moving, or maybe a group of substances. For example, there's the glucose transport protein in the liver that carries glucose into the cell, but it doesn't carry fructose, which is a structural isomer. So very specific. And again, think enzymes. The glucose transporter causes glucose to pass through the membrane 50,000 times faster than it would if it were just diffusing by itself. So a very effective transport or carrier protein. You can see here the glucose protein in the model here showing how it's actually binding to the protein, and that is allowing it to change shape through the middle and allow that glucose to then pass. We'll talk a little bit more about co-transport, but that is also occurring with this type of carrier protein. So that's why you actually see these little aqua-colored molecules there. Just as an interesting side note, a lot of human diseases are actually resulting from different malfunctions in the transport system across cell membranes. And one of those is cystinuria, which happens when there's an absence of a carrier protein to transport cysteine and other amino acids across the membrane of kidney cells. And so what happens when this occurs is there becomes a buildup of cysteine and amino acids in the kidney itself, and actually stones start to form. There is almost like a calcification of these little protein packets within the kidney cells, and that's what a kidney stone is, or it's actually one type of kidney stone that occurs. And of course, if anybody knows someone who's had kidney stones, that's a very painful condition due to the lack of these carrier proteins. Okay, so now we've been talking about diffusion when we're moving from high concentration to low concentration. So now if we want to go reverse, if we want to go from a low concentration to a high concentration, we're moving against the concentration gradient. So there's a need for energy for that to occur, and that's called active transport. So in order, you can kind of think of proteins in this case as being a protein pump. So like a little machine that's actually using energy, just like any mechanical machine would. There's an energy source for mechanical machines. In this case, with proteins, the energy source is ATP. And so ATP allows that energy to pump the molecule, whatever it is, against the concentration gradient. So again, we're going, in this case, from a low to the high concentration. So this is a summary of the 3 types of transport across the cell membranes with the help in 2 cases of proteins. In the one simple diffusion case, the transport occurs without any assistance. Just goes right through the cell membrane. Now, another 2 ways that transport can occur across the cell membrane is in the case of very large molecules or large numbers of molecules. If there is a need for that, that's when we use endocytosis or exocytosis. So a cell will use vesicles or vacuoles to enclose something, either large molecules or again large numbers of molecules, in order to either get rid of them outside of the cell, which would be exocytosis, or to bring them into the cell, which is endocytosis. And that process takes the form of, actually, I'll go back here to show that. What is important with these vesicles or vacuoles is that they are made of phospholipids as well, so that they fuse easily with the cell membrane. They're like material, like molecules, so they're able to fuse and become part of the cell membrane themselves. And so that's what you see happening here. As it becomes part of the cell membrane, it actually just naturally is going to open up and release what was inside. And that can go in either direction, either out of the cell, as in this case, or into the cell as well. So when it goes into the cell, again, it's called endocytosis, and there are 3 types of endocytosis. Phagocytosis is when you fuse with a lysosome to digest something. Pinocytosis is when water is enclosed. And receptor-mediated endocytosis is when there is exactly what it sounds like. A signal is received by a receptor on the cell membrane, which signals the cell membrane to enclose the signal molecule along with others and bring them into the cell. So these are all ways that things are brought into the cell. Okay, for the rest of this lecture on Chapter 7, I'm going to focus on movement strictly across the cell membrane, which will have us looking deeper into diffusion and osmosis. But before I do that, to break this up, I have a joke for you, or a riddle. Why can't plants escape prison? It's because their cells are surrounded by walls. Okay, diffusion. So diffusion again has to do with the second law of thermodynamics, which is talking about entropy, where there is a movement towards disorder. And that's what drives diffusion. So basically, if you have a high concentration of something, there is, according to the second law of thermodynamics, a movement or a draw towards more disorder. So there is a spreading out or a spreading away from the highly concentrated area. So this governs not only physical sciences, but also biologic science. And so we see an example of it here. This is molecules of dye, and this is a membrane that is allowing has pores that allow the molecules of dye to pass through from their high concentration to a lower concentration. And then this is again a review of the 2 major types of transport across the cell membrane. So we've talked about passive transport, which includes simple diffusion or facilitated diffusion. And then we have active transport, which is actually with the help of ATP energy, and it's diffusion that's occurring in the opposite direction. It's going against the concentration gradient from low to high, and it uses a protein pump. So I won't spend any more time on simple diffusion because I think you really have that at this point. But we'll stop here for a second with facilitated diffusion. So each transport protein, so we talked about the ones that actually act as carrier proteins, but even those that make channels in facilitated diffusion are specific to the substance that they actually move. So, for example, aquaporins are the channels that move water across the cell membrane. They only will bring water across. Although I have to make a little bit of an adjustment or addition to that statement because there is more recent thought that some sugars actually pass through the aquaporins as well, and actually play a role in helping the water move through. So that's something for future research. Okay, and then with active transport, again, you're moving with the help of a protein pump against the concentration gradient. Okay, so I don't think we need to spend much more time on that. This is a summary again of that slide we had before on the three different types, passive versus active transport. But what we haven't really talked a lot about so far is co-transport. So in co-transport, a membrane protein couples the transport of two different solutes, two different molecules. So one solute will be actively transported by passively diffusing back through the transport protein. And that passive diffusion gets coupled with active transport of another substance against the concentration gradient. So again, looking at, I've told you we'd come back to that glucose transporter protein. That is one that does co-transport. So in plant cells, we have an accumulation of hydrogen ions occurring through photosynthesis. And so there is a natural diffusion of those protons across the cell membrane through this glucose transport protein. And so that natural passive diffusion, actually facilitated diffusion because it's through a protein, occurs. And as it occurs, it actually energizes this protein to then be able to take glucose molecules and bring them against the concentration gradient in the other direction. So it brings one way and takes the other. Um, Actually, in this case, it's not going against the gradient. I'm not going in the opposite direction, but oftentimes they do go in opposite directions. For example, with the sodium-potassium pump would be another example of co-transport. But in this case, they're both going in the same direction, so they're coupled. So it can be coupled in either direction. Okay, and we talked about endo and exocytosis, so I'm not going to spend more time on that, except again to point out that you need to know these three different types. So again, phagocytosis. Phagocytosis literally means cellular eating. So think of it as engulfing something to eat it. Penocytosis is cellular drinking. So it has to do with transporting a bunch of water molecules. And receptor-mediated endocytosis is when something is triggered by a molecular signal. So an example of that one is human cells use receptor-mediated endocytosis to take in cholesterol for use in membrane synthesis. So cholesterol, as you know, travels in our blood, and it actually travels in the form of a low-density lipoprotein, which is a complex of protein and lipid. And these lipoproteins, when they arrive at a cell wall, they act as a ligand, which you will read more about in Chapter 11. But a ligand is like a signal molecule that binds to a receptor on the cell membrane. So as you were making your membranes, you saw that you were building proteins that were embedded, and attached to those proteins were either a lipid or a carbohydrate. And a lot of times what those molecules are like catcher's mitts or receptors for a signal molecule, which we call a ligand. And so when, in the case of cholesterol, when the LDL gets one of these bound to the membrane protein, then a signal is sent to the membrane to actually begin this endocytosis process. And so by doing that, it actually will then bring in all of those Lipoproteins, low density lipoproteins, will then be enclosed by